Ganz herzlichen Dank den zwei Klosterschülern, Manuel Bucher und Kilian van Meuden. Sie haben zuerst das Prelude Nummer 1 Kilian, glaube ich, von George Gershwin gespielt und jetzt zusammen die zwei ungarischen Tänze von Johannes Brahms. Herzlichen Dank für euer Auftreten. Herzlichen Dank, Ab Christian, für Ihre Worte und herzlichen Dank, Landamann Niklaus Bleiker, für die ersprießlichen Wünsche, die sicher Früchte tragen werden. Und jetzt kommen wir zum Ernst des Tages. The Power of Genetics, from Flies to Humans. Es ist Professor Ernst Hafen. I introduce him to you. Ernst Hafen received his philosophy doctor from the Biozentrum at the University of Basel in the year 1983. After being a postdoctoral fellow at the University of California in Berkeley from 1984 to 1986, he joined the Zoological Institute of the University of Zürich as an assistant professor in 1987. After becoming associate in 1994 and full professor in 1997, he served as the director of the Zoological Institute from 2004 to 2005. In 2005, he accepted a professorship at the Institute of Molecular System Biology at the Faculty of Biology at the ETH Zürich. Ernst Hafen made several contributions in the field of developmental biology and cell biology. He received various awards and served on the editorial boards of several journals and scientific committees. We are very happy to have you here, and you are a newly elected member also of the Foundation Council. Welcome here. I give you the word. You have the floor, Professor Hagen.
So that the Akademie Engelberg has decided this year to choose the subject of personalized medicine for their main discussion forum. Because I do think, and what I will try to do is I'll try to tell you how I, as a geneticist, got interested in human genetics and personalized genomics, and how I'm convinced that this field will have a lot of opportunities and challenges for the future, for society, for healthcare. And it is time to talk about this now because the change is happening right now. But we're still, I think, in a position to start this dialogue, and this is a great forum to do that. So I will tell you first, very briefly, what we're talking about, and I'm taking a very simple analogy. You hope to excuse me for that, about what the genome really is, what do we understand when we talk about the genome. And then I will tell you how, for the last 20 years or so, we have used these small model organisms, flies and other model organisms, to understand biological processes by using genetic tools, and how this research from many, many different groups in the world has contributed to a great understanding of the basic principles of how cells divide, how cells die, how organisms develop, and how organisms die. And that it is now time that we move on from this sort of analysis of the basic processes, but we want to understand how these basic, highly conserved processes actually govern the different appearance and the different susceptibilities of each and every one of us as we are today and in medicine. So these will be the three parts, and I will tell you why I got so excited about personalized medicine when I visited the Congress several years ago in the United States. Now let's see what we can advance here. Yes, we can. So this is okay. So we're talking about the genome, and the genome is in each and every one of our cells. We inherited our genetic information from our father and our mother in one copy each, and it's built up in 46 chromosomes, 23 from the mother, 23 from the father. And the chromosomes itself are just long stretches of this DNA molecule that you all know, this sort of ladder, double helix, which has these little base pairs, which are the letters of our genetic information. And if you count all the letters that are present in each and every one of our cells, this is about 6 billion letters in each and every one of our cells. And this is you cannot really see what this is. So for the 175th anniversary of the University of Zurich, a group decided, amongst Michael Hengartner, to actually show what this actually is. And so they decided to take from the internet the entire sequence of just one chromosome of the 46 chromosomes in each of your cells, and they chose the X chromosome and decided to print that out on a laser printer and make a book out of it. And I'm showing you this book right now. This is also a very important book. This is the sequence of the X chromosome. This is 145 million letters. And if you look what this looks like, I can open it. It's 600 pages of print like that, which you obviously can see. These dark lines here are the information that we know something about. Those are the genes that encode the instructions to make specific proteins. All this sequence here is sequence that lies within the genes. It probably has important information. We still know very little about this. Now, you don't see this. I have brought the magnifying glass, so after the break you can look at this, but I've actually zoomed into this. So this is the page again, as you've seen it before. We zoom in higher, we zoom in again higher, and we zoom in higher. And now you see all these 4 letters, the ATGs, in 154 million copies. This is the sequence of the X chromosome. Now, this is completely ununderstandable for us as it is here. I can't do anything with this sequence. The interesting thing about this sequence, however, is that if we compare the sequence of 2 individuals from each other, the sequence is not identical. We are not identical twins. Usually, we're different. But we're different in terms of the genetic sequence in 1 in 1,000 letters. 1 in 1,000 letters. That's all the difference, genetic difference, that we have between individuals. So if you take a big book like War and Peace, and you would think that every 1,000th letter is a misspelling, you would probably not even notice it, and you would go on reading it. The content would not change. However, here, this 1 in 1,000 differences has quite a dramatic effect and lays the foundation for the differences between individuals. Now, it is always said, so the genome is not the blueprint of what we are. The genome is the foundation of how we're going to develop, and this depends on all the environmental influences that we're exposed to. And so it is much more that we have to learn from the genome as the basis to where we are. So this is the genotype, and this is the phenotype. And in order to sort of symbolize that, I like to take a very simple analogy that I actually got from René Lago, the head of the pediatrics clinic in Zurich. He's now retired. He explains the difference with air balloons. Each of these air balloons is different. Let's look at these 2. Or let's look at the green one and the red one. These are different in color and probably also in size. And this is basically what the genetic information tells you. But it's hard to actually see what kind of color, what size, and what form this is. So in order to see that, you have to blow these air balloons up. And the blowing up of the air balloons is sort of the realization of the potential of what you can do with the genome. So that's the education, that's the nutrition, that's your exposure to environmental cues. And so it's immediately evident that you can blow up these balloons not enough. So if you do that, you will not even see that this will be a heart. Or you can blow it up too much, and then it explodes. And these are all the environmental influences that reflect what can be done with the genome. But something that you can never do, by blowing into this red balloon, you will never change it into a green balloon, and you will never change the form. So this is basically the basis that is in our genome. And so the challenge for personalized medicine and for biology and life sciences is to find out how these very small differences, 1 in 1,000 letters in this book and 46 copies of this book, will translate in the phenotypic appearance and in the susceptibility to diseases or the reaction to medications. And this is the topic of today. Now, we got into this problem as geneticists, not by using humans, but we wanted to understand the basic principles of how do you build a very simple organism, a fly. And you know these flies from your fruit basket. Those are Drosophila melanogaster, small flies, easy to grow, rapidly proliferating. They have a 20-fold smaller genome. They have the same genome, but it's 20-fold smaller. And we can do genetics by introducing type mistakes, base pair changes randomly and see what happens. And in this way, we're knocking out individual genes, individual information, and then find out what they do. And from that, we go back and see what this gene could be doing. The human geneticist, you will hear from Uta Franke, does exactly the same thing with hereditary diseases. You identify the gene that is affected by the disease, for example, in chorea Huntington, and then you learn something about how this disease occurs. So we can do exactly the same thing because these sequences are conserved. So ultimately, the question that we want to answer with our fly work is, how do you get from genotype to phenotype? There's a long way, but you'll hear during the conference many ways of how this can be approached. Now back to the flies. So this is our model organism. What we're doing is we're looking at flies that are now really identical twins. They have identical genomes, and then we are randomly putting errors into their genome sequence and selecting the ones, in our case, the ones that are smaller than normal or bigger than normal, because we're interested in how does our genome define the size of the organism, the size of the balloon in the analogy. So we can do that very rapidly. We can do that for hundreds of thousands of flies and actually find all the genes in the genome that have something to do with body size control. And what we find is, and what we found over the last 30 years, is that the genes that we identify are also genes that we have in our bodies and that do actually surprisingly similar things in our bodies. So they're very old. So we find, we don't have to go into detail, we find, for example, that the insulin signaling pathway regulates growth and size in flies as it does in humans. So flies have insulin, and they have insulin for precisely the same reason that we have insulin. It is a signaling system that allows the body to know whether you have taken up nutrients or you have not taken up nutrients. And so many of the components that have been identified here and arranged in these pathways, this is a single cell with insulin on the outside, acting on a receptor and then acting over a complicated cascade. A complicated cascade to give instructions to individual cells. So this is one cell, this is the nucleus with its genome in there. And so this is the complicated pathway. This is highly conserved in all organisms. And as we also know, it has something to do not only with growth, but it also has to do with another process called aging. So flies have a limited lifetime, but this lifetime, to some extent, if you treat them the same, is also genetically determined. So, for example, our small Chico flies that you just see, so they have an infunctional gene, which we call Chico, and therefore they're smaller. But those small flies also live longer. These are the wild-type flies that live about 40 days. And if you just have a single defect in this one gene that weakens the signaling through the insulin signaling pathway, you get flies that live about 60 days on average. So we have a pathway, and this is true for many, many processes, that the same pathways control different phenotypes in animals. This goes to a very old evolutionary problem, and the problem is that organisms that survived today and from which we are the descendants had to develop strategies to survive under conditions where there was no food. So all the strategies that allowed them to survive for a longer of time were transported through evolution. The fact that you have too much food because you're in a supermarket or in a snack bar is a relatively recent problem, and so evolution has not really coped with that. That's why obesity is such a big problem compared to caloric restriction. And in all these animals, it has been shown that caloric restriction, so if you just don't feed the animals as much as they would normally eat, about 60% of what they normally eat, they live on average about 30% longer. Because each cell has learned to protect itself from this not availability of food, they activate all these stress protecting programs, and that makes the animal live longer. Evolutionarily speaking, this will have saved the species from extinction because it can wait until there is new food sources. I'll show you a movie that shows you how deep ingrained this aging process is from a colleague of mine, Cynthia Kenyon from UCSF, and this is another model organism, the worm Caenorhabditis elegans. It's a small worm, lives in the soil. And we're looking here at these worms. You can grow them on petri dishes, and you see they live only about 13 days, so they're great for aging research because they die so quickly. And you can do many generations. Well, you see there is no difference between this normal worm that has a perfect genome and the one that has the identical genome, but it also has a mutation in the insulin receptor gene. So like Chico, it cannot transmit the insulin signal so well. So this worm thinks it is starved, even though it has enough food. So after one day, you don't see a difference. But now let's look at 13 days. The wild-type worm, so at the end of its lifetime, his colleague is already dead. And this guy here is really a very old worm. He cannot hardly move. He can move his head a little bit. If you compare that to the worm that has this defect in the insulin pathway and thinks it is starved and therefore activated all these stress protecting programs, this worm will live twice as long, but not only will it live twice as long, it also looks much more useful. It crawls around still at the same age. So a mutation in the genome controls longevity. So these basic processes, I said, are highly conserved between organisms. And the aging or nutrient sensing pathway is just one of the examples. It is conserved from yeast to worms to flies to mammals. And the same genes have been always retained their function and allowed this nutrient signaling in these different organisms. So this is great. So for the last 20 years, we have learned quite a significant amount from studying yeast, worms, and flies for human aging. And many of the mutations and pathways that we learn probably are involved in human aging as well. And we know very well that human aging, different people age differently, and then there's probably a hereditary component to that. But before I show you a very impressive example to this case, I want to also reflect on what that really means. The fact that our analysis, this sort of knocking out single genes analysis, has provided us with these fundamental pathways, but it has also just said, well, you know, flies are like humans, which they're obviously not. So all these pathways that we are studying have been here 600 million years ago or older. They have been present 600 million years ago in a sort of ancestor to the common ancestor between vertebrates and invertebrates, for example. And you see that this trilobite here had all the pathways that we already discussed now. Cell division, nutrient sensing, growth control, etc. All these processes have already been established, and they have been used over and over again in the different species to implement the differences. But what we now, knowing all these conserved pathways, these pathways must be used in different ways to come out to the differences in different species or between different individuals. These different individuals all have exactly the same pathways, they have the same genes, but they're different in 1 in 1,000 nucleotides. And this small difference affects the activity of these individual pathways, that many of them we know, and we have to find out how these different activities affect these different appearances. One example in terms of aging is this remarkable woman, Jeanne Calmet, the oldest woman to date. She died 1997 with 122 years old. When she was 115, she stopped smoking. But she did not stop smoking because she felt it was bad for the health, but because her eyesight was turning weak and she couldn't light her cigarette herself. And she felt it was too embarrassing to have someone light her cigarette for her. So she stopped smoking. So clearly, there are differences in the aging population, and this is what we're trying to find out. Now, so in our own research, we're going to move more into sort of looking at populations of flies, wide populations of flies, which have a similar sort of very small variation between the different individuals. And we're trying to find out how these small variations affect our conserved pathways. But I realized the problem with flies and with mice and with worms is that they have a great disadvantage from humans. First of all, they're obviously not humans, and humans go to the doctor and tell them about their phenotype, or they can write about their phenotype. Whereas flies and worms and mice, we have to find out everything about them. We have to torture them to find out their phenotypes, etc. And this is a difference with humans. They can actually help to contribute to this understanding. Each of one of you can contribute to this understanding. And I realized the importance of this about 3 years ago when I was at a conference at Google, where I met many of the speakers actually that are going to speak in this session, and that really left me transformed and see the potential for also human genetics to directly contribute to this understanding. One of the technological advances is that these are the costs for sequencing an individual genome. 10 years ago, for the first genome, this cost $3 billion to sequence the genome. So this is these 46 or 23 books that you've seen there to determine the sequence of each individual. It was $3 billion. Now, in March 11, it was slightly over $10,000. And you can extrapolate this sequence, and you can see that within 2 years, sequencing your personal genome, your own genome, will be less than $1,000, probably 100 Swiss francs or 200 Swiss francs. So this technology is happening, this is here, these sequences will be available, you can order these sequences. So we have suddenly the possibility to compare our genome, the genotype, with our own phenotype. And having multiple such comparisons, millions of such comparisons, will give us the power to understand more how you get from the genotype to the phenotype. This is one aspect that I learned there. And the second aspect that I learned, I met with 2 companies that are also going to be represented here, at that time startup companies, that actually utilize this information. And 23andMe, I'm going to tell you more about this, actually offers this genotype analysis, genome analysis, personal genome analysis as a service already now. Not the entire sequence, but basically 1 million points along the entire 46 chromosomes. It's like 1 million bookmarks that have been correlated with different diseases. And Patients Like Me, Jamie Haywood is here, he's a founder of Patients Like Me, is basically a social network for chronically ill patients where they can put in their information about their illness, their medication they're taking, and share it with other people that have the same disease. And of course, this is an important information resource to find out how medication works. Jamie will talk about this. I should point out that in neither of these companies I have any financial interest, but they were eye-openers for me. So when I came home after this conference, I said to my family that the Christmas present, my Christmas present for my family in 2008 was their complete genome analysis. And so this is my wife, Eva, Raphael, the oldest, Timmy, middle, and Robin, the youngest. They're all over 18, and 2 of them study biology, and 1 is a civil engineer. So they said, yeah, that's great. Why don't we do that? So this is what you get when you then order this kit from 23andMe, a little box, and in there is a vial in which you put spit in it. It has a lot of DNA and cells in there. You send that back with a barcode, not with your address, so your address is not linked to your DNA sample, and you get an access into your own account on their website. And I'm going to tell you now what this looks like, because it was an interesting experience. We didn't learn much that we didn't know before. But the idea of 23andMe, and the idea is that each of us can now contribute to research by having his genome and talking about his phenotype, its diseases, etc., and thereby contributing to the understanding of how you get from this complicated book to our phenotypes. So I'm going to take you very briefly through my genome. So this is the 23andMe website. I've been logged in as Ernst Hafen here in my account, so I can share my data with any one of you, and I've shared it with several also here in the audience. But this is the raw data. So here you have your 46 chromosomes. We've been looking at the X chromosome here. This is the book. You see 454 million bases. It has 1,300 genes, and at that time, 27,000 SNPs. Those are those markers that they assay. So it's not the entire sequence, but they assay those markers. Now, you can even download this data. This is like printing out this book. It doesn't help you at all. But I want to show you what the sequence actually looks like. So I typed in insulin because we were talking about insulin. We all have an insulin gene that makes the insulin hormone, and I just chose this. This is now on chromosome 11 here at the tip. And here you have 2 such markers within the insulin gene. Those are the name of the markers, the positions along the entire sequence. And you have here variable regions. You can have, at this position, this is a region that is variable in at least 10% of the population. You can have, people can either have a C or a T. In this position, it's an A or a T. And obviously, because you get a sequence from the mother and from the father, you have 2. These, incidentally, are my father and my mother. And so they were CC at this position, my mother also, so the only chance I could have is that I have CC. If I were CT, then I would have to look a little bit more closely. And maybe talk to my mother. But at this position, again, there is AT. My father and my mother are heterozygous, and I have a quarter of a chance to be AA. Again, this is just how it works. It doesn't tell you anything. But the same markers have been used for over 1,000 clinical studies where people have recruited 1,000 patients with type 2 diabetes and 1,000 patients in a control group. And they did exactly the same analysis. And they asked which of these versions correlate more with the people who have the disease or those that don't have the disease. And those are these genome-wide association studies that Uta Franke is going to tell you about more in her talk. And from this information is aggregated on this website and compared to our genetic information. So you get this risk information that you have, so type 2 diabetes, for example, let's take this as an example. The confidence here means that several independent studies have been done with several, 1,000 patients each. And from that, it was calculated if you have this particular genotype with all these variants that I have, the average risk in the population is 23%. My risk is slightly lower at 16%. So again, what do you do with this information? That's a question that is going to come up over and over again. I was not so excited about this, although it is very well done, I think, even for a geneticist and for non-geneticists. It then explains you what type 2 diabetes is and what these markers mean. But I'd like to share a more recent finding that just came out accidentally from this analysis. And you can be carrier for a disease because you need 2 copies of the disease to manifest itself. And hemochromatosis is one of these relatively common diseases that has been linked to a certain one of those markers. And we knew from our genetic analysis that my wife is carrier for this disease. She doesn't have it. She's heterozygous. She has only one marker. And I'm not carrier. So our sons, 2 of our sons are carrier of the disease and 1 is not. And the oldest one married 2 years ago, and I convinced my daughter-in-law to take the same test. And she immediately called me up and said she was carrier as well. It turned out. And so if Raphael, the oldest son, would be carrier, the son, our grandson, first grandson, would have a 25% chance to have the disease, be homozygous for the disease. She was very happy to learn that Raphael was the only one of the 3 sons who was not a carrier of the disease. And here is Seraphine, our grandchild. So he may be a carrier, but he certainly is not at risk of getting this particular disease. So it has some very interesting information in there. But the main point of this is that we can participate in research by providing our information, our phenotypic information, to 23andMe and have them associated in much larger studies than those clinical studies that we've talked about. And I'm happy to say that several papers have been published, research papers have been published by 23andMe based on the data. They have now over 100,000 such genomes in their database. And so this leads to new associations and a better understanding of specific traits or diseases. So this is a new way of doing research. It's a research where everyone can participate, everyone can contribute. It's almost like Wikipedia for medicine. Everyone who knows something can contribute. And having the genome as a unique identifier and being sort of the basis for that is really the big advantage. So that's why I thought it was important, that we thought that it was important to start talking about this, because the DNA sequencing technologies are available that very soon everyone will be able to do that. And this is the opportunity, so DNA sequencing will be ubiquitous. Phenotype-genotype correlation with public participation, one calls this crowdsourcing. So a combination of 23andMe and patients like me, as you will see from later talks. And I'm convinced that personalized medicine will improve, personalized genomics will improve personalized medicine, as we've heard yesterday from Esther Dyson's talk, in terms of more prevention, lifestyle changes, and also drug doses. But of course, it also generates challenges. How we deal with all this data? Am I happy with the fact that my genome data are in a company? And what happens to my data when the company is no longer financed? What is the toxicity of the information? This is something Robert Greene will address soon. What if I learn something about a disease that I didn't know about? How can I deal with this? Is this really toxic for me? Can I adjust to that? And what about the acceptance in the public? Der gläserne Patient oder der gläserne Mensch. How are we going to deal with this? And once again, the genome is not your destiny. It is the form of these balloons. How you blow them up, how you that is really what we can do. And we can change this still. And the more we know about ourselves and our predispositions, the better we are off. But these are topics that will have far-reaching consequences in healthcare, in society, and that's why I'm so happy that we're talking about this now. So, towards the challenges, just very briefly, we may talk about this. There is a Bundesgesetz über genetische Untersuchung bei Menschen called GUMP. It's the equivalent to GINA in the United States that already now prevents the use of genetic information in health insurance, employment, in life insurance until a certain amount of money. If I want to get a health insurance for 500,000 Swiss francs, they will ask me whether I've ever done a genetic profile. And they can ask me that. And then I say, yes, I have done that. So they will look at my 23 and want to look at my 23andMe profile. Not that this is a problem, because they will send me to a doctor anyway, and the doctor will ask me how much I drink and how much I smoke, and whether I have any sort of severe diseases in our family, which is still, at the moment, more informative than the genome sequence, but that's what the law says. And the REVEAL study, this is something how toxicity of genetic information, whether it is toxic, that Robert Green will talk about that in extenso. And then finally also the fact that the young generation, and I'm talking about students, are usually very much more open to this kind of sharing of information. And this is a survey I did half a year or a year ago with 335 first-year biology students after talking about human genetics. And I asked them, would you, for 200 Swiss francs, donate your genome information and your phenotypic information to medical research? And almost half said yes. Some said less, said no, and a significant number was still undecided. But I think there is a fairly large acceptance among the younger generation. So in summary, I think we're going to talk about all these topics in great detail and from real experts, not from fly doctors like myself. But I'd like to just briefly summarize what I've said. So we have talked about the small differences that are the basis for the appearance and susceptibility to diseases, the foundation for that. I've showed you that these biological processes that govern our cells are highly conserved, are very old. So we're not talking about a pill for each and one of you that is completely different, because you all have the same pathways, but you may need different combinations of drugs that suit your type 2 diabetes. And what we want to find out is how small changes in the genome generate these phenotypic differences. This is a humongous task, which needs really studies with millions of people, and this is not done by classical publicly financed studies. This needs participation of the public. And the syncing DNA sequencing costs permits this participatory research if it is then approved. And so that's why I think it is important to start a dialogue about this technology right now, and that's why we're here. Thank you very much for your attention.
Thank you very much, Professor Hafen, for this fascinating presentation. I think we have several minutes for questions.
I need to be clear, I'm a scientist. I believe that science is the only way, decent way to learn. But nevertheless, I do not believe that every possibility provided by science is good to be used immediately by society. Yesterday, we had Mrs. Deason, and she explained first that apparently, and I'm surprised about that, she thinks that genetics is not so important. We'll not learn so much out of it. I think this is not correct. We are going to learn a lot. Indeed, we are going to learn as much as in this case. Imagine, I know nothing about you, but imagine you have a twin, a real twin, and I know everything about your real twin. I will know a lot of things about you. That's not the state of present-day genetics, but it will come. Now, Mrs. Deason was explaining yesterday that privacy is something that it's an idiosyncrasy of the 20th century. Before it didn't exist, now it's already gone. I'm afraid with that. And indeed, I saw Mrs. Deason coming in front and speaking to us with a very well-studied position. She knew what she wanted to show, not only with a text, but with a posture. And indeed, all of us are dressed. We don't like to be nude. It's a fundamental human right to show ourselves as we want. Now, opening our genes is showing us nude, terribly nude. Are we prepared to that? I think it's good to be true, to show the truth, but we need to be prepared. Goethe, oh, I don't know. No, Victor Hugo said, oh, sorry, I do not get it. Le soleil, la vérité est comme le soleil, tout le monde la loue, mais personne n'ose la regarder en face. We should learn how to look in front to look truth in front, but it will take time. Merci beaucoup.
Thank you very much. I think you raised a very important point. I'm happy that you say that you don't agree with Esther Dyson that genetics is not so important. I hope I convinced you that genetics is important. But it is not all. And it is precisely this discussion that we have to lead. What does the genome of our cells really, does it present us nude? Or what is the sequence really meaning for us? You know, it's like this sort of uninflated balloon. It's different from you as you are now. And so we have to understand, and we can only do that by learning more about the genome and talking about this early on. And I really want to start this precise discussion about this. I don't have the precise answer for that. That's why we're here. But to start that, not like with the Genschutzinitiative, where we had the green gene technology, basically after the fact we started discussing, and there were, you know, the gain mice was out there, and everyone said, no, this is not for us, and this is because big companies are behind this financial interest. We don't want that. And we have won, luckily, or turned down the Genschutzinitiative, thanks to a lot of efforts from many of the people in the audience. But the discussion was started too late. And I think we want to start a discussion now from a forum like this that has no sort of direct links to companies, etc., and from a university. And that's why we're starting this. We want to start this awareness campaign about personalized genomics now, to talk about precisely these points.
Thank you for the talk, very good. My question is, one of the fundamental rights as patient is the right not to know. So how would you have managed that in your family when one of the sons said, no, I don't want to know? Like in Facebook, you can more or less access to your data to whom you want. Is that, I think we have to find a solution to that, because that would be one of the fear of many people.
The right not to know, I think, is an important right, and in our situation, with 23andMe in my genome, for example, the associations with some markers that give you an indication whether you have a high risk for Parkinson's or not, where there are a few genes that have a larger effect. There, you were asked several times, do you want to know this? And you can just say, no, I don't want to know this. And I think this is important. This option has to be there. But this does not prevent me from telling in a survey that I have no the family history of Parkinson's, even if I don't know what I have in my own genotype. That I think is important.
Thank you. My name is Christian Larsen, medical doctor, and in orthopedics and biomechanics. Now, as far as I understood, the individual information is very subliminal. You know, you said 1 out of 1,000. And the same thing can be observed in biomechanics. Let's say, for example, a tennis player like Nadal or Roger Federer. It's on, if you do statistics from 100 different tennis players, the main, let's say, characteristics are the same. But underneath there is a very subliminal individual signal, like a fingerprint, and this can be, allows every movement to identify the player. If once you have the fingerprint, you can identify, out of 1,000 different movements, you can identify it. Now, as a medical doctor, I'm confronted with two models in science. One is the natural science, and one is the quantum physics. And now and it's like a controversy and like two different worlds. And now there starts to be some evidence. For example, the birds, how they manage to find their orientation when they are flying south through quantum effects in their inner ear. And the idea is that biological systems are like very powerful amplifier of very subliminal signals. Now the question is, do you think that the linking the genotype and the phenotype will bridge the two worlds of science, from quantum physics to natural science?
No, I don't think so, because again, your genome is not deterministic. So it will, even if we understood everything how this is translated, it will not be deterministic. It will probably determine the different behavior, Nadal versus Federer, probably. There is probably a genetic component of how they do their forehand, I could imagine. We don't know how that is. These phenotypes are very complicated. They're not caused by, you know, the forehand gene. There are thousands of genes that cooperate, thousands of gene variants that cooperate with each other. And understanding these interactions is a big challenge for the future, but it will never be deterministic.
But that's not what I meant. I meant the opposite, that each individual is unique.
Well, obviously.
Yeah, that's obvious.
Yeah. Each individual is unique, but even if you took 1,000 balloons and you blow them up by different people, they will not be identically blown up, because they will have different amounts of air in them, even if you count the individual molecules. So there will be difference. But they will be different based on the genotype, but also on the environmental influence. So this will be a discussion that will go on, I think, for the rest of the meeting, which is, I think, very important. We have to come back to this. Yeah?
Can I make a comment? It's not a question. It's a simple comment about hemochromatosis, and it's not directed towards you. It's directed towards eventual doctors in here. Now, I'm homozygous for hemochromatosis, and I suffered from all the symptoms for almost 20 years, and none of the professors of medicine at the University of Zurich were able to make the diagnosis until I did that myself, because, of course, I have studied medicine many years ago. All symptoms have disappeared, and I'm absolutely confident that between 5 and 25% of all diabetes of maturity onset are due to hemochromatosis. A large number of cardiac irregularities are due to hemochromatosis, and a lot of arthritic pains. All these are classical symptoms of hemochromatosis. It should be excluded when any of these symptoms appear, and it's a simple test. It's called ferritin. Thank you.
Thank you very much.
I would like to make a remark
to the previous questions about the right to know. In our discussion with the young scientists yesterday, we took another perspective of this issue, naming at a given stage or in a given situation, the society probably would like to have the right to know. For example, the case of very aggressive people who are in prison, who have, let's say, deviation, the behavior deviation. When is it possible to release them, or is it necessary to identify them genetically that they are not released, as we saw many cases in the recent months?
But I think I mean, it's a valid question, but I mean, this is a question for society that we always had, and it will be a long time before you can make a genetic test to predict the behavior of an individual such that this can be the basis for releasing someone from a prison. I think we are far away from that at the moment. We just have to see how limited our knowledge is, but how we can build on that.